Hello and welcome to the 6th episode of the Innova Biosciences webinar series. Today's webinar, A Beginner's Guide to the Principles and Applications of FRET, will discuss the powerful technique of FRET, or Fluorescence Resonance Energy Transfer, and the various ways in which this is utilized in biotechnology. Great. So our speaker today is Dr. Andy Lane. He will introduce you to the basics of FRET and its applications, which range from the construction of so-called tandem dyes to increase the range of reagents suitable for use in flow cytometry, to assays designed to identify molecular interactions as part of drug discovery research programs.
Great. Thank you, Claudina, and good afternoon or good morning, everyone, depending on where you are. As Claudina said, I'm going to talk about FRET today, and I'm deliberately going to keep things really quite simple. It would be quite easy to get into a lot of detail about FRET and to confuse you perhaps with quite a lot of equations, etc. You may be pleased to know there's only one equation in this webinar today, and I'm certainly going to keep this for beginners to give you an idea of the wide applications of FRET and see if we can encourage you to make more use of that as a technique. So a little bit about the background of FRET to start with. FRET stands for Fluorescence Resonance Energy Transfer, and it was actually described more than 50 years ago. I think it's fair to say that over the last 10 to 20 years, the availability of new dyes and also of hardware in the form of detection technology has resulted in a much wider use of the application, especially in biomedical research and development. In very simple terms, FRET is dependent on distance-dependent transfer of energy from a donor molecule to an acceptor molecule. That results in the loss of fluorescence from the donor and a gain of fluorescence of the acceptor molecule. And you can measure either of those, and we'll talk about that in a little bit more detail in just a moment. Most importantly, FRET is therefore an extremely powerful tool for identifying molecular interactions. It can be used in a variety of base techniques, including microscopy, flow cytometry, and ELISA. There are a whole range of conditions that influence FRET, but the two most important ones are distance and spectra, along with dipole orientation. If we think about distance first, the donor and acceptor molecules must be in very close proximity to each other, and that's generally considered to be between 10 to 100 angstroms, i.e. less than 10 nanometers apart. In terms of spectra, the absorption spectrum of the acceptor molecule must overlap the emission spectrum of the donor, and I'll show that diagrammatically in a couple of slides' time. And perhaps slightly less often considered is that the dipole orientation of the donor and acceptor molecules should be approximately parallel. Now, these are the main conditions that influence FRET. There are many others, but given that we're talking in a beginner's session today, I won't take any time in thinking about them. Let's look at distance in a little bit more detail. Assuming other criteria are met, the most important factor is the distance between the pair of molecules. And as I mentioned earlier on, I'm only going to include one equation today. Forster showed that the efficiency of FRET is dependent on the inverse 6 distance between the pair. And this is the equation where E equals RO to the power of 6 over the sum of RO to the power of 6 plus R. Now, RO is the Forster distance, and that is the distance at which 50% of energy is transferred. R itself is the actual distance between the pair. Now, as with many equations, it's probably easiest to actually put that into context. And in practical terms, this means that the efficiency of FRET is extremely sensitive to distance. If the molecules are moved 10 times further apart, the efficiency of the reaction falls by 5 million-fold. So I think on that basis you can see how distance is one of the primary influencing factors on FRET. The next condition, which is of particular importance, is the spectra of the donor and acceptor molecules. Now, if you take this diagram, which is in cartoon format, we have an absorption spectrum of the donor and an emission spectrum of the donor. If you excite the donor, it will then emit light at a longer wavelength, i.e. at higher energy. This will overlap, hopefully, with the absorption spectrum of the acceptor dye, which will then emit energy at its own emission spectra. For a donor and acceptor pair, we are looking for an overlap here between the emission of the donor and the absorption of the acceptor dye. The bigger the overlap, the better in terms of FRET efficiency. However, what you don't want to happen is that the absorption of the acceptor dye overlaps with the absorption of the donor dye. If that is the case, you get direct activation of the acceptor dye, which will lead to confusing results, to say the least. If there is any overlap on a small scale, you may be able to deal with that with filters in your instrumentation, which I'll mention again a little bit later. FRET itself can be measured in a number of ways, but effectively you're doing one of two things. You're either measuring the increase in fluorescence of the acceptor molecule, or you're measuring the decrease in fluorescence of the donor molecule. In terms of measuring the decrease in fluorescence of the donor molecule, that is simply because the emission of the donor molecule has been taken up by the acceptor molecule, and therefore you don't see that as an emission event. There can also be advantages to expressing the increase in fluorescence of the acceptor together with the decrease in fluorescence of the donor as a ratio. That means the measurement is then not dependent on the actual concentration of the molecules, but as a ratio between them. In another widely used variant called acceptor photobleaching, the acceptor is deliberately photobleached so that it can no longer be activated by donor emission. In such cases, after the photobleaching, you'll see an increase in donor emission, and that actually shows that FRET was ongoing before the photobleaching event took place. Now, this is one of the slides within the webinar that could be an awful lot longer and could probably be 10 slides in itself. The number of FRET pairs that are used is increasing very rapidly, and I've really put a very small selection here. Picking out on some of these that are in this selection are perhaps some older molecules, fluorescein, which can pair up with the acceptor tetramethyl rhodamine. Then we have the group of fluorescent proteins that are generally used being genetically encoded into molecules. So, for instance, CFP or cyan fluorescent protein with yellow fluorescent protein, and blue fluorescence protein with green fluorescence protein. Then you have a group using the lanthanide rare earth metals, europium and terbium, which again are a very popular set of pairs. Europium very commonly with allophycocyanin, or more recently with dilite 650, and terbium with a number of acceptor molecules including phycoerythrin and dilite 550. You also have the smaller organic molecules that can be used as donor and acceptor pairs, for instance dilite 488 combining with dilite 550, and a very common pair of Cy3 together with Cy5. As I mentioned, this is a very small selection of the number of FRET pairs that are actually available for use. In terms of experimental design, there are a number of general things to consider. Obviously, one of the key factors to consider will be what base application you're working in. In terms of base applications, I mentioned applications such as ELISA and microscopy earlier on, and obviously your experimental design will depend upon which of those that you're using. Obviously, perhaps the primary thing is to select an appropriate donor and acceptor pair. These are going to be based on their inherent suitability as a pair, as we've already talked about, and also, to a certain extent, on your equipment. Obviously, there is no point utilizing a pair for which you don't have the right excitation wavelengths, or even the correct wavelengths to be measured. You should choose suitable filters to maximize excitation of your donor, whilst minimizing any direct excitation of the acceptor molecule. Consider the concentration within your system. Are you actually likely to have enough detectable events? Remember that what you're measuring is the actual interaction between the donor and acceptor molecules. So simply having high concentrations of donor and acceptor in itself isn't necessarily enough to generate a signal if the interaction is very low. And remember that it's the interaction that you're measuring, and your experimental design may actually show that there is no interaction there, and therefore there will be nothing to measure. Consider which of the molecules might be better as the donor and which as the acceptor. Unpaired donor molecules have the effect of diluting a signal, whilst unpaired acceptors have very little effect. Therefore, if you have a choice, use the molecule at the lowest level as the donor. Making the choice of donor and acceptor early on is obviously key to your experimental design. You may have to be in the sense of preparing particular reagents. So if you need a particular color or dye for your donor and acceptor, you don't want to be changing those during the course of a set of experiments. So think carefully about how you can design and choose your reagents at an early stage. If possible, use a control for the FRET reaction itself. Now, you'll need all your normal standard set of negative and positive controls for the experiments that you're running, but if you can have a control to prove to yourself that the FRET reaction is working and that you can detect it, that is also quite useful. A very simple control of this nature is to use a primary antibody labeled as an acceptor and a secondary antibody labeled as a donor. When you add the secondary antibody into the system to bind to the primary, you should then get very high efficiency FRET reaction, and you can use that as a positive control. I now have just a few slides of cartoon examples of some ways in which FRET can be used. In this first example, we're looking at the interaction of two molecules and of detecting that interaction. Now, these molecules, for instance, might be a drug binding to an active site on a protein, or perhaps an antibody binding to a target molecule. On the left-hand side here, we have the drug, which is labeled as a donor, and here we have the protein molecule. In this set of conditions, the two are apart, and you will be seeing donor emission. So if you're activating within the absorption spectrum of the donor, you'll then be measuring emission of the donor and no emission from the acceptor. If biological interaction takes place, i.e. this molecule binds to the active site, FRET will then occur and you will be able to measure acceptor emission. This could also be exactly the case if this is an antibody and this is the target. This is exactly the same sort of reaction that is going on. In this way, you can screen for drugs that may bind to particular sites on molecules as one example of a FRET experiment. In almost the opposite experiment, you can actually look for compounds that block the interaction of two molecules. So here on the left-hand side is exactly what we saw on the right-hand side of the previous slide. We have a molecule that is bound to the active site of this protein, and we're seeing FRET occurring and emission of the acceptor molecule. If you can block that biological interaction, perhaps with another drug, you can then see that FRET will stop, the acceptor emission will be lost, and you will be having as a readout increasing levels of donor emission. In this way, you can screen for drugs that will block biological interactions. In another example, you can actually use FRET for detecting the co-location of molecules on or within cells. This again is a very common application for FRET. In this cartoon, we have a diamond-shaped cell surface molecule which has been detected with this antibody. We have a triangular-shaped cell surface molecule and a square-shaped cell surface molecule. And you can see quite clearly that the diamond and the triangle interact quite closely at the cell surface. And with two specific antibodies to those molecules, if you label them simultaneously, you will be able to see the FRET reaction taking place. This will show you that you have a very close proximity of these molecules at the cell surface. You note that the diamond and the square are not associated, and in this case there is no FRET reaction occurring. This is looking at the cell surface in this cartoon, but you can easily look within the cells, within particular organelles, etc. In a slightly different example of FRET, you can use the system indirectly to detect molecules such as proteases that may actually cleave proteins in half. So in this diagram, we have one protein, we have a donor molecule at one end, and we have an acceptor molecule at the other. Whilst this protein is intact, the FRET reaction is occurring. Now within a particular system, you may be screening for protease molecules that can cut this protein in half. Those proteases might be from a library that you're screening, or they may be expressed within, for instance, a cell culture supernatant following a particular stimulus. If the protease is present and the protein is cleaved, the donor molecule and the acceptor molecule are moved much further apart, and therefore the FRET reaction stops. In this way, you have an indirect measurement of the level of the protease in the system. In another example shown here, you can look for the detection of a conformational change within a protein. In the upper conformation, we have a protein in which the donor molecule and acceptor molecule are a fair distance apart and no FRET is occurring. However, on a stimulus that leads to a particular conformational change, the donor molecule and the acceptor molecule are brought together and FRET can be measured. Again, if you're measuring a particular stimulus that you know induces that conformational change, this gives you a measurement of the level of that stimulus. In a particular example of FRET, tandem dyes can be constructed for use in flow cytometry. Now, for many people, when they use tandem dyes in flow cytometry, they're probably not thinking of a FRET reaction at all. Many monoclonal and polyclonal antibody manufacturers will conjugate their antibodies to tandem dyes such as RPE-Cy5 and RPE-Cy7. And these work on the basis of FRET. On the left-hand side of this diagram, we have the RPE molecule being stimulated by laser light at 488 nanometers. And RPE will emit light at 575 nanometers under these conditions. If you link small molecules, in this case size 7, to the RPE, it will then undergo a FRET reaction, so that if you stimulate RPE at 488 nanometers as before, the emitted light, which is at 575, is transferred directly to the size 7, which then emits itself at 775 nanometers. The effect of this is that, in effect, you have a new color of dye, and this is particularly useful for multicolor flow cytometry. The advent of tandem dyes perhaps 20 years ago was one of the major factors in moving flow cytometry from 2 colors to the multicolor experiments, perhaps of 12, 15 or even 18 colors that are possible nowadays. In terms of preparing reagents for FRET, there are probably 2 main ways of approaching this. The first way is to use a genetically encoded dye, such as the fluorescent proteins, GFP and YFP. Obviously, these can be encoded genetically, but you have one thing to bear in mind here, is that you will only get 1 copy into each molecule. The other approach is to use chemical conjugation of target molecules to incorporate the appropriate dyes. These might be antibodies, other proteins, or even perhaps oligonucleotides. Be aware that there can be some advantages to using antibodies against the target molecules if they're available, rather than using genetically encoding. The main reason for this is that you can get higher levels of dye incorporation into the antibody as opposed to the single copy that you will get if you incorporate something like GFP into a protein. In terms of preparing reagents, the lightning link system from Innova Biosciences provides perhaps the world's easiest set of kits for labeling proteins. This is a simple 1-step process which literally takes 30 seconds hands-on. If you want to work at very small scales, kits are available as low as 10 micrograms in terms of a conjugation amount, and these provide 100% recovery of material. The Lightning Link kits are available in a very wide range of formats, including a number of those that we've talked about in the webinar so far today. Obviously, these would include the tandem labeling dyes. So all of these tandem dyes in a Lightning Link format are presented so that you can conjugate the tandem dye directly to an antibody or another protein. There are also some of the other fluorescent proteins that are familiarly used in FRET reactions, such as allophycocyanin and R-phycoerythrin. Within our rapid kits are also some of the newer organic dyes that can be used for FRET reactions, including the Cy3 and Cy5 pair, and also the commonly used acceptor Dye-Lite 650. If you're looking to use Lightning Link to prepare reagents for FRET, there are just a few things that you need to know. Lightning Link conjugations themselves, as I mentioned, are very simple indeed, but it's best to have the protein in the right format for the conjugation to work effectively. The format really needs to be a concentration of around about 1 milligram per mil, or preferably a little bit higher. You also need to make sure that your protein is pure. And I've noted here to say that make sure after purification certain proteins haven't been put back in again afterwards. In terms of antibodies, a number of antibody manufacturers will use proteins such as bovine serum albumin to stabilize antibodies after purification. In that case, that will interfere with the conjugation reaction and it is best removed. In terms of buffer formulation, your protein will be okay in most common formulations, but make sure that amines, such as glycine, are truly absent. Therefore, if glycine has been used in a purification process, make sure that it has been extensively removed by dialysis. Make sure that thiols such as DTT or mercaptoethanol are also absent. Tris is okay at relatively low concentrations. The Lightning Link kits that are available from Innova are optimized in terms of their instructions for antibody labeling, but that can easily be adjusted to label other proteins. Basically, if you know the size of your protein, you can calculate how to use Lightning Link for your application. And obviously, we'll always be able to help you out with that calculation if you need some support on that.
So a big thank you from Andy and I today. Thank you for joining us. We hope you enjoyed it and that you'll be able to join us again for future webinars.